We're going to be taking notes on the cell cycle. We're going to be using a long sheet that we're going to be doing a little folding and cutting. And you're also going to need a regular writing utensil. So 4 color markers, a regular writing utensil. So first we have to set up our paper. So it's printed. We've got this kind of center area and then the 2 edges. We're going to fold these over along the line. So fold in. We're going to open it back out, and where we have the dotted lines along the edges, we're going to cut. So once you're done, the whole thing, we'll fold inwards and have 6 flaps. First color, you can pick whatever color you like. That's going to be kind of your labels and title color. So we're going to label this whole thing at the top, the cell cycle.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material.
So for our cell cycle, I'm going to take my regular writing utensil, and I'm going to start by drawing my first cell on the top left corner. And inside that cell, there is a nucleus. And I want to label nucleus. And then I'm going to pick one of my colors. I'm going to draw some centrioles. The other 2 remaining colors, I'm going to draw some chromatin inside the nucleus. It's just some squiggly lines for right now. Same title color, and make sure you're still inside the flap here. I'm going to put my hint in between. I'm going to open up, and behind I'm going to put the name of this stage, and I'm going to use my title color here. This is Interphase. And there are a couple important things happening here. We've got replicating DNA, which means to make a copy. We've got cell growth. And we're preparing to divide. Now, I want to pause a minute, and we'll close that back up. We're actually going to flip to the back side for just a minute. And I'm just going to do small in the top corner there. I'm going to draw just a big circle. And this is going to be a pie chart that shows me how much time the cell spends in each of these parts of the cell cycle. And I'm going to make my pie look kind of like a 2 o'clock. And then that small slice, I'm going to divide a little bit less than 1/4 of it. A little bit less than 1/4 of it. I'm going to label that so I know what that means. Time in cycle. And I've got 3 stages happening here. So interphase, we just drew. That's this big part here.
And what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and it's replicating its DNA and carrying out some of its cell processes. That's what it spends most of its time doing.
The bigger of the smaller slices, does that make sense? So this bigger one here, that's mitosis. And then the smallest one actually has the longest name. It is Cytokinesis. So let's flip it back to the front. Now we're going to get into mitosis. So mitosis is actually several steps. Here's a quick visual overview of mitosis in some real cells. So I'm going to take my title color and I'm going to put some stars in the top corner, then both the next ones, and then the bottom left. Because all 4 of these, so you should have 4 stars when you're all done, all 4 of these are going to be parts of mitosis. And you can see when we open it up that there's some lines inside drawn around these little parts. So we're going to open it all the way up and we can see our lines inside here. And right in the middle, I'm going to write what this is. I'm going to put a little bubble. It's mitosis, which is division of the nucleus. So we call it nuclear division. So interphase is stage 1. Mitosis is stage 2. And the bottom here, this will be cytokinesis. Stage 3. Our first step is called prophase. So our hint is actually partners or pairs. So I get my hint color. So my partners. And we're going to draw using our regular pencil our cell. It's about the same size as it was here. But our nuclear membrane is starting to disappear. So I'm drawing a dotted line. And inside of the nuclear membrane, we have the chromatin folding up. And it's forming special shapes, these kind of X-shaped. And these X-shaped things are called chromosomes. So they're made of chromatin, it's just folded up into this special shape.
Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
Centrioles, which were already in pairs, starting to head away from each other, head to opposite sides of the cell. But they want to stay in touch because they're partners. So they stay connected, these special little fibers. They're called spindle fibers. So let's open this up and write what's happening here. Got the name of this part of mitosis called prophase. Now I'm using my writing utensil. So we have spindle fibers form. The nuclear membrane disappears. And chromatin condenses to form chromosomes. Here's a picture with a few cells pointed out going through prophase at different points. So one has the nuclear membrane gone. In this picture, can you find the one going through prophase? If you guess this guy in the middle, you'd be correct. We're going to take a closer look at chromosomes, so flip it over to the back. And we're going to draw a chromosome. So it's got 2 sides connected by something in the middle. And both sides are made of DNA. And I'm going to label this, doesn't matter what colors. So both sides are made of DNA folded around some proteins. And each side is an identical copy of the DNA. We call it an identical rod of DNA. And that has a name called a chromatid. Highlight it with the color I chose. So each side is a chromatid, an identical rod of DNA, and the middle that holds the 2 chromatids together is called a centromere. So this whole thing, once again, both sides together, is called a chromosome, and that's formed when the chromatin condenses or folds up around those proteins. Here's a picture of someone's chromosomes. It could be yours. Back to the front. Let's see what happens with these chromosomes. So up here, they were partnered in prophase, first part of mitosis. Now, second part of mitosis, our hint is middle. Looking at this picture, can you guess why? So I draw my cell, a little more oval shape maybe this time. Nuclear membrane, remember, long gone. And my chromosomes now line up right along the middle of the cell. The centrioles are on opposite ends. We call those the poles. And their spindle fibers are now connected to the middle of my chromosomes. This picture is a computer rendering of chromosomes lined up in the middle, and they're so long you can see they're overlapping. That's the blue in the middle. The green is the spindle fibers. And this is just a cell under a regular light microscope, and we can see the chromosomes lined in the middle. Open that up and put that information about mitosis. This is metaphase. Use my title color, and we're going to put what happens in metaphase. We've got chromosomes line up in the middle. And the spindle fibers connect to the centromeres. Our hint for this one is going to be apart. And now my cell is stretching out, so it's a little bit longer than it's been. Stretching out almost like a peanut here. Centrioles still on the opposite ends, but now they start to reel in those spindle fibers. They're pulling them into themselves. And as they do, they break apart, that's the hint, the chromosomes, and it looks like a streamer being pulled behind someone when they're holding it in the middle. Behind. Let's open that up. And I get my title color. This is anaphase. Where the centromeres split apart and the chromatids get pulled to the opposite end by spindle fibers. And then we also noticed that the cell stretched out. Here is those green spindle fibers pulling the blue chromatids to opposite poles in anaphase, and once again under the regular light microscope. Alright, so almost done. We got anaphase. Now our last box for mitosis is 2 nuclei in telophase. So now we're going to have 2 nuclei formed from all those parts. And our cell is starting to look like it's pinching. And we have nuclear membranes come back. And the chromosomes and the chromatids, they're done. So the chromatids start to stretch out. And centrioles are just kind of hanging out. So they're done with their job for now. So they're just floating around in the cytoplasm for the moment. So let's flip that open. Got telophase. So now we have 2 new nuclei. So our 2 nuclear membranes form. And the chromatids stretch it out. So then our final stage of the cell cycle after mitosis is cytokinesis. So cytokinesis, stage 3, starts, overlaps with telophase. So that's why the cell starts to pinch in. But by the end of cytokinesis, what we have is cells, plural. So we have 2 cells, not as big as the original cell was. They're baby cells. We got 2 nuclei in there. And the new cells get about half of the organelles and each get their own nucleus. So they each get, we'll just draw the centrioles to represent the rest of the organelles being split. And the chromatin is totally relaxed back again. So it's back to that original squiggly line. So we open it up, and cytokinesis, we're going to have 2 identical cells, and we call them daughter cells, and each daughter cell has about half of the organelles. That's cell parts, remember. So half the mitochondria, half of the ribosomes, half of the ER. And 2 identical nuclei so each daughter cell has full copy of the DNA. So the only other thing I think we should do is go back to our prophase, metaphase, anaphase, and give some number just to make sure we remember which order those come in mitosis. So prophase first, so I'm going to put a little 1 next to it, then metaphase, then anaphase, then telophase. And on the back, I'm going to put a little memory trick This is just the first letters of each of the phases, and I remember I, P, mat, C. And PMAT, that's all mitosis. And that is the cell cycle. How we go from one cell to two identical daughter cells.